Hello, my lovely anatomist and physiologist, Michele here. Thank you for joining me for another Special Senses video. Our topic for today is gustation. Gustation, remember, is our sense of taste. As we look at each of our special senses, we are keeping up with the sense organ, information on the sensory receptor, as well as that afferent pathway. So that's really what we're looking at with this particular thing. When we talk about gustation, our sense of taste, we all know that that's happening in the mouth, and we see that specifically located on the tongue. Now, I wouldn't really say that the tongue is our sense organ here, but this is where our sense organ is located. Now, you know, because you're humans, and I'm sure you all have tongues, you know that your tongue is bumpy. And we talk about these bumps, this is a plural word, papillae. So if you're talking about a single bump, it would be papilla. Now, papillae are these, this is a term we'll use quite a lot. We've used it before in the course. It's talking about like a finger-like projection. And these papillae, these bumps on your tongue, can have two jobs. They can be involved in manipulation of food, or they can also be involved in taste reception, or I'm going to write here taste sensation. And when we talk about the papillae, we have four types. So I'm going to start first with the filiform. This one is located on the tip of your tongue. And this one is more about friction or manipulation of food. So here I like to think like this is what's letting me like really scoop up that ice cream off of that ice cream cone and get that into my mouth. Then we can list our fungiform, which is going to do both food manipulation and taste sensation. We can do our foliate, which is going to be involved in taste. And then the last one, let's actually put a star beside this one. You can call it a circumvallate, or you can just call it vallate. And this one is along the back of the tongue. And this one is really, I'm going to just write the word taste really huge. The circumvallate is the one that's really involved in taste sensation. So how can we know whether the papillae is there as a bumpy projection to help us manipulate food, or whether it's there involved in taste sensation? If it's involved in taste sensation, so that would be the fungiform, the foliate, and the circumvallate or vallate, then it contains as part of its anatomy the taste bud. And I think this is what I will really classify as the taste organ. So if we go back to our chart that we started looking at with that very first video, and we have our special sense, and then we have our sense organ. I think for gustation, I would call that the taste bud. Then when we talk about the taste bud, let's first list out the types of cells, and then we'll try to do a little drawing here. So we have 3 types of cells. We're going to get a basal cell, which is our stem cell. Now you should remember this from Anatomy and Physiology 2 or Part 1. Any kind of basal cell or stem cell or germ cell, these are all names describing a cell that is mitotically active. And mitosis, remember, is cell division. So a basal cell is a cell that's able to constantly divide and produce transitional cells. The transitional cells, the transitional cells I'm going to call immature cells. So these are the cells that were produced by the basal cell or the stem cell during mitosis, but they're not quite ready. They're not quite mature enough. When they're fully mature, we call them a gustatory receptor cell. And so this is the cell that's actually able to, let's say, be stimulated by a tastant. So remember in olfaction, we had an odorant, which was the chemical that bound to the G protein coupled receptor and stimulated the olfactory sensory neuron. In the case of our gustation, the gustatory receptor cell is also doing chemoreception. And so again, it's a chemical that's going to stimulate it, and we call that chemical here a tastant. So the gustatory receptor cell is our specialized receptor cell, and we see that it's key to the structure of our taste bud. So we can go back up to our chart, and where we see specialized receptor cell, let's write in here the gustatory receptor cell. Okay, so let's see what we can do about a drawing of our taste bud. Now, what we see as part of our papillae anatomy is we have our papilla. So I'm going to call this here the papilla. And then we get this like taste pore. And so this opening here is called the taste pore. And so as we eat food and we manipulate it with our tongue and we smush it around, we're starting that process of digestion, but we're also getting those tastants into this taste pore. And what we will see here is we will have, let's do our gustatory receptor cell in green. And so we're going to have these cells that have, let me make it smaller. We're going to have cells with microvilli, so that's extensions of the cell membrane. And then we're going to do several of those in a line, but let's leave some space. I'm just going to do a few. I'm not going to make the whole taste bud here, I don't think. The gustatory receptor cell is going to be synapsing onto a sensory neuron. So we can do our sensory neuron here in yellow at the base of the gustatory receptor cell. And then I'm just going to grab my black and I'm going to put a few of these transitional cells in between. Remember, the transitional cells are immature. They don't have the microvilli. They're not synapsing onto the sensory neuron. They're not quite ready. And then we're going to have at lower down here, we're going to have some of those stem cells as well. So we can label that green one our gustatory receptor cell. We can label our transitional cell, which is our immature gustatory, and then we can have our stem cell. And then at the bottom, we're going to have our sensory neurons. And this whole sort of picture then is making up that taste bud. And when we talk about our sense of taste, we're tasting salty, we're tasting sour, We're tasting sweet, bitter. These are classic ones. I think we're mostly familiar with these. We also have a taste sensation that's classified as umami or savory. And then there is another one called oleogustus. And this one we'll see is like bad. This is like rancid fat. So this is a very unpleasant taste sensation. As we look at our afferent pathway, we're going to see that salty and sour are going to both be using ion channels as part of their stimulation of the gustatory receptor cell. But then we'll see the sweet, the bitter, and the umami using a G-coupled protein receptor. And I don't actually know the mechanism for oleogustus, so we won't be going into that in a lot of detail. So as we move along, what we're going to be ready for next then is looking at our gustatory pathway. And we're going to do it for each of those taste sensations in a little bit of a separate way. All right, y'all, so let's start with the taste sensation of salty. And when we are talking about salty, what we're really talking about is our hydrogen ion being dissolved in our saliva. And we can actually also just make a note here that sour, when we talk about sour, what we are detecting here is, mess that up, y'all. I got super distracted. Salty is the sodium ion, right? Y'all were like, what is she talking about? Sour is the hydrogen ion. So really we're talking about something that's acidic as tasting like sour. And so then the mechanism of action is the same, whether we're talking about salty or sour, they're just going to be moving through different ion channels. So in each case, what we see is that the ions will be dissolved in saliva. This dissolved ion then is going to enter an ion channel in the gustatory receptor cell. So remember those gustatory receptor cells had those microvilli. Those are those extensions. That's just silly. Those are just those extensions of the cell membrane, right? And so that's just increasing surface area. And so in those microvilli, in those extensions of cell membrane, we're going to have some of these ion channels. And so we have salt that can move through, sodium ion, we have hydrogen ion that can move through, and that's going to depolarize the gustatory receptor cell. That's our signal, right? And so this signal then is going to stimulate the sensory neuron. And those neurons are going to collectively make up the following cranial nerves. So we're going to see the facial nerve, which is going to be the anterior 1/3 of the tongue, or we're going to see it stimulating the glossopharyngeal nerve, which is going to be the posterior 1/3 of the tongue, or it might be the vagus nerve. And the vagus is going to be at the very, very, very extreme posterior part of the tongue, almost moving into the pharynx, which is the back of the throat. And one of the things that we'll see is that the vagus is involved in the gag reflex. And we won't see gag reflex so much with the salt or the sour, but we will when we get into like either bitter or oleogustus. Okay, let's look next at our sensations of sweet, which is a monosaccharide, or bitter, which is something that's usually alkaline. Collectively, we can talk about those as alkaloids. Often bitter is a bad flavor. So for instance, coffee is very bitter. This is why people like to combine it with sugar and milk, right? And older people enjoy black coffees typically more than younger people, and this could partially be the reduction in taste sensation that does come with age. So bitter is often associated with things that are poisonous and can be part of that gag reflex. And then we mentioned umami as a savory flavor. And so this is coming from an amino acid called L-glutamate. And so umami flavors are often found in meats, but you can also have things like tomatoes and eggplants that have a lot of umami. Some cheeses like mozzarella has a lot of umami. And these sensations are all G protein coupled pathways. So that means that the monosaccharide, the alkaloid, the L-glutamate is serving as a first messenger binding on that gustatory receptor cell, activating a G protein, which then is like hollering, hey parent, somebody's at the door, to trigger then your second messenger. So in this case, we're just going to be generic and say the taste ant binds to the receptor in the gustatory cell. And we'll see that this can either activate or deactivate the cell. And we see that deactivation more with bitter taste sensation than maybe with the monosaccharide or the umami. And we're not activating or deactivating the cell, but rather the G protein. The G protein then, if it's activated, is going to activate the second messenger. And so here we don't have a specific one. It can be either the cyclic AMP or it can be the calcium ion. And then depending, this is going to either depolarize. So if the G protein was stimulated, then the G protein is activating the second messenger, then we're going to typically see a depolarization, but we might get a hyperpolarization with those bitter flavors, right? And here we have the same scenario that we had a minute ago. So we can actually just grab this information here and we can add it right here. So we're still traveling along the same pathway with those nerves. Now, the thing that I didn't finish, which I'm just seeing, is the processing center. So for both, we're going to say our processing center for both, we're going to have our processing center here is going to go to the brain stem, goes to the thalamus, and then we're going to go to the gustatory cortex, which is located in the insula lobe. And let's make sure to add that in our previous so that we're clear. That that's the same here as well. Okay, so that finishes us up on the gustation. If you have questions, let me know. And stay tuned. Up next is hearing. And as always, take care of yourselves and each other.